Hello everyone, my name is Paul Roberts and I want to tell you a most amazing story. It's a story of intrigue and illicit affairs that happened over 200 years ago, with changes in names, and it involves the Industrial Revolution in England and military service in India, the last Zulu War in South Africa, and then the greatest diamond rush that ever occurred, and then the gold rush to the Witwatersrand in Johannesburg. And the identity of the hero of the story and of his parents and grandparents were all discovered using DNA. This is the story of my great-grandfather, Richard Charles Roberts. After the first 40 years of genealogical research into the story of Richard Charles Roberts, all we really knew about him was that he was born in Birmingham around 1858 on the Bullring or near it, and that his last name had been Roland, R-O-L-A-N-D. And then we knew that he served in the military in India, the last Zulu War in 1879 in South Africa. Then he married his first wife, Mary Agnes James, and had 5 children, and she died young, and then he married Maria Sophia Brousseau, the mother of my grandfather, Walter Edward Baden Roberts. Richard finally died in 1929 on the gold mines in Johannesburg. We had searched repeatedly, taking the cold case file off the shelf again and again, but never found any Roland child born in Birmingham around that time. In fact, we didn't find any R-O-L-A-N-D Roland born in England. Found no marriage records of the Roland parents being married. We could find nothing of Richard Charles Roberts' English ancestry, although we presumed he was 100% Anglo-Saxon, of a family probably living Middle England for centuries. Then finally, about 10 years ago, DNA became more and more used in ancestry research. And I said to my father, this may be our last resort. This may be the only way we find out who his ancestors were. If we send in my dad's DNA, and if we could match with a family in Birmingham with the last name Roland, then we'd be able to trace them and trace my dad's history. And my dad said, fine, let's do it. So we sent off my dad's sputum in a little vial, which Ancestry.com sent to us, and we sat back for 6 weeks waiting for the results. We were in for a big surprise. But first of all, I just want to review a little bit about DNA and explain it to those who may not fully understand. This is an important summary slide for anyone interested in having their DNA tested for ancestry purposes. Just remember that here is a cell. Our entire body is made up of cells, and the cells contain little organelles inside. There's the mitochondria, which produce energy and contain their own unique type of DNA. And there's the nucleus, which contains the DNA for the entire structure of who we are, what we look like, and in many cases, what we think like. The DNA is all unraveled. It's just like a bowl of spaghetti, these long strands of multiple millions of code. It does not normally look like this. The pictures we see of DNA like this is when the cell is ready to divide, then the DNA starts coiling up like a roll of yarn into what are called chromosomes. And humans have 46 chromosomes. There are 23 pairs, one from the mom and one from the dad in each pair. And there are 22 autosomal pairs and 1 pair of sex chromosomes. This would be a female because they're identical, XX. This would be a male with an X and a Y. But there are about 3 billion base pairs. And those are the coding units, 3 billion segments of code. And the interesting thing is that 99.8% of that 3 billion is identical. So every human on the planet shares the exact same 99.8% of their DNA, because that determines that we do have eyes, that we have eyes on either side of our nose, that we have a liver and the function of our liver, that we have kidneys and the function of our kidneys. The entire makeup of humans is coded in this DNA. So 99.8% of us is identical. It's only in 0.2% of that genome that we vary. So every different human being you see on the planet is only varying in 0.2% of their total DNA content. But that 0.2% is about 10 million base pairs. So still a massive amount of code. The DNA tests only look at about 500,000 base pairs, so a 20th of the total variation in DNA amongst humans. This is the typical image we see of DNA that we're all familiar with. It's called the double helix. That's the structure of the molecule. It's kind of like a ladder that's been twisted. And this structure of the molecule was first described in the early 1950s by two scientists, Watson and Crick. So it is a relatively new science. Although back in the 1860s, the first paper was written about genetics and the inheritance of traits. That was by Gregor Mendel, a monk living in Austria, and he began studying genetics in peas. And since then, genetics has become a major area of study, and so much more has been learned all the time. But the basic molecule is DNA, which does look like this, sort of, and here's a symbol of it in this side of the diagram. And the strands are the items of code. There are 4 base pairs in DNA: thymine, cytosine, adenine, and guanine, and that's their chemical structure. But each molecule of DNA contains this strand of code connected with the corresponding strand on the other side. Cytosine will only connect with guanine and not with thymine or adenine. Thymine will only connect with adenine and not with cytosine or guanine. And that is what determines our code, the sequence of those 4 bases on the DNA molecule. When the DNA is going to make a protein in the body, it unravels, the 2 parts of the helix come apart, and that code is revealed, and that code makes the proteins. So the proteins of structure in your body, the enzymes that control all the reactions in your body, are all produced directly from the code from the DNA. While I was waiting the long 6 weeks for my dad's DNA results to return, I started looking at his ethnicity, as that is the first thing I would look at when the results arrived. We know that ethnicity is not extremely accurate as we would predict, because recombination occurs in the chromosomes when they replicate. But roughly, my dad should have been 50% Eastern European. My dad got half his DNA from his dad and half from his mom, 23 chromosomes from each. And my dad's mom, my grandmother, got half her DNA from her dad and half from her mom. And my grandmother's father, his parents both came from this little town in what was then the Austro-Hungarian Empire called Altkloster. It's now Kazakh or something in Poland. And my grandmother's mother's family both came from a little town in northern Czech Republic called Supikovice. So that means my grandmother, her blood was entirely Eastern European, Austro-Hungarian Empire. Then on my grandfather's side, his mother and her parents both came, Frederick Brousseau and Maria Prufrock, both came from the Uckermark region of Germany, actually from Strasbourg, a little town up there. So not one of them had a bit of English DNA in them. And then there was Richard Charles Roberts from Birmingham. So we were pretty sure that my dad would have a significant amount of English DNA, maybe up to 25%, and then 25% German and 50% Eastern European. Finally, the results arrived on Ancestry.com, and I excitedly opened up to have a look at them. When you get onto your results page on Ancestry, you see you have three options. Origins, which is your ethnicity estimates, DNA matches, and through lines, and I'll speak about them a lot later. But currently, my dad's ethnicity estimates are 36% Germanic Europe and 26% East Europe, Russia. So in combination, that's the Germans and the Austro-Hungarians, and 6 other. I was interested in what are those others. So to find that, we click on Discover Your Origins. When I first did that, this is what I found. And so this is where my dad's DNA came from, this part of the world, as we expected. Didn't know why all that dark blue was down there. But it said French, German, 36%, British and Irish, very small, 3%. It should be 25, shouldn't it? Eastern European, 23%. Southern European, 15.5%. That's Italian, Greek, etc. 15.5%, there shouldn't be any. But this all added up to that German Austro-Hungarian percentage we were expecting. So we didn't know what to make of this. At a later point, I sent my dad's DNA into 23andMe to check the results there. And again, it showed Northwestern European 58.4%. That's a combination of French, German, and Eastern European. And then Southern European, 7.7%. Came up with only 3% British. So there was a lot of unexpected Southern European Italian. This may be a scary looking slide reminding us of days in high school biology, but it's very important just to remember a few basic principles of mitosis and meiosis. Mitosis here on the left, that is the normal cell division that occurs in every cell of our bodies every second. Cells of our skin and the mucosa lining our intestinal tract are being replaced continually as the surface layers slough off, and every cell in our body, as it divides, as I mentioned before, every nucleus contains all the chromosomes, all 46, and the new cells have to also contain 46. So mitosis is normal cell division starting with 46 chromosomes. In this example, there's 6, and ending with 46 chromosomes, identical DNA contact to the parent cell. In this example, it shows there are 3 pairs of chromosomes, 3 blue, so this cell got 3 from the dad and 3 from the mom. All the DNA does, those chromosomes, they do not pair up with their partners. They just individually replicate themselves. And that's a detailed process I won't get into. But each chromosome replicates itself. So now this cell has double the amount of DNA, each chromosome duplicated, and then those chromosomes just line up in the middle and split. Each half goes to each end of the cell and the cell splits. So it's very simple. The DNA just doubles itself, makes a identical copy of itself, and then the cell splits. That's mitosis, and that's going on in our bodies all the time. Meiosis is what is important to us in genealogy because it explains why, although we get 50% of our DNA from our dad and 50% from our mom, we do not get 25% from each of our 4 grandparents. Our dad got 50% of his DNA from his dad and 50% from his mom, but we are not getting 25% from each grandparent. And that sounds a little confusing, but it's because of the way meiosis occurs, which allows randomization so that every one of the gamete cells will be uniquely different. Again, in this example, there are six chromosomes, 3 pairs. In meiosis, this is the process of division that produces the sperm and the ova. So it's going to produce those cells, the sperm and the ova, with half the DNA of the parent cell. And then they will combine with the sperm or ova of the other parent and produce the baby. So in this case of meiosis, those pairs of what are called homologous chromosomes pair up together. So chromosome 6 of the father will find chromosome 6 of the mother, and they'll snuggle up side by side. And chromosome 21 of the father will find chromosome 21 of the mother, and they'll snuggle up side by side. So they all pair up. Those are called the homologous pairs pair up. And then there is a random exchange of DNA between them. Those long strands of DNA, they are divided into sections containing all the genes, thousands and thousands of genes. And one gene, on chromosome 16, if it happens to be the gene for eye color, it'll be in a specific position on chromosome 16 of the father, and in the paired gene from the mother, it'll be in exactly the same position. And randomly, these are switched between the 2 chromosomes, completely randomly. And this randomization occurs, so DNA is shared between the 2 chromosomes, and then the chromosomes migrate to the poles of the cell, and the cell divides, and you end up with each of the gamete cells, either the sperm or the ova, has only 3, in this example, 3 chromosomes. In humans, it would be 23 chromosomes. Not 23 pairs, but 23 individual chromosomes. And then that sperm or ova can unite with the gametes from the other parent. And it's that randomization, which is completely random, at exchange of DNA, that makes every sperm and gamete different. And you may at some point get a lot of the father's DNA on one chromosome and very little from the mother. But in the next gamete on the same chromosome, you may get mostly DNA from the mother and very little from the father. So when the offspring are produced, you may end up with a lot of DNA from one grandparent and very little from the other. And that's why we do not get 25% of our DNA from each grandparent. So back to my dad's DNA specifically, in his ethnicity estimates, we had found an amazing amount of Southern European Italian DNA, which we totally didn't expect. And this could only have come from Richard Charles Roberts or Roland from Birmingham. So the next step was to look at my dad's DNA matches. For those of you not familiar with Ancestry.com, when you've sent in your DNA and you get your results, this is what the matches page looks like. This shows you it's my father's DNA, and there are various groups you can look at. You can look at those with common ancestors, those are people you have not viewed before, and you can sort the ancestors into groups, or the matches, into groups, and that's very important. The matches are all listed in their closeness to you, or in this case to my dad. So the first two are me and my brother. We share 3,400 about centromeres of DNA, about 50% of the DNA tested, and by the amount of DNA shared, the computer predicts we must be sons of Vernon. It shows that yes, we do have a public tree and we have a common ancestor on that tree. Many people have public trees, and that's very helpful. Some people have private trees which you cannot see, but you need to message them and ask them if they will show you their tree. And many people send in DNA and don't have a tree at all. But pretty much anyone who has sent in DNA that matches with the DNA we've sent in, they'll be listed down here, and it's going in descending order of closeness to the person tested. So the first person that came up was Glenn. And I've added a little note that he is the great-grandson of Uncle Ernie Winter. But when he first appeared, we didn't know who he was. We were told he's a second to third cousin, somewhere in that range. So we share great-grandparents or great-great-grandparents. And the amount of DNA he shared was 288 centromeres. He did have a public tree, and it tells us we have a common ancestor. We know that now. Originally, that wouldn't have been there. The next match is Craig. He's a descendant of he's a grandson of Uncle Ted Winter. Again, a second to third cousin, sharing a similar amount of DNA, and this proceeds. The next person is DW, and it turns out he is a grandson of one of Richard Charles Roberts' daughters that he abandoned when his first wife died in South Africa. He also shares 256 centromeres of DNA and has a tree. So there were no surprises so far. What I decided to do was to lump these people into categories. Over on this side, if you click on that little pencil, you see it gives you the option to add a group. And I decided to make the Wachowski or Winter group green. So each of these people who were related through my grandmother and her siblings, I marked green. They're on the Wachowski side of the family. So I could immediately remove them from my concerns with Richard Charles Roberts. Dennis, we knew, was on Richard Charles Roberts' first wife's side, so originally I marked him red in the Roland Roberts group. Then we came down to Brousseau. We know that my father's grandmother on his father's side was Maria Sophia Brousseau. She was Richard Charles Roberts' second wife. So I marked the Brousseau group, this dark purple color. So now we're down to still second and third cousins sharing 178 centromeres of DNA. But then suddenly there's a Sophia and a W.K. last name, and they're second to third cousins. And this K family were shared matches with Richard Charles Roberts' family. To solve problems in our tree, it is essential to start grouping our matches into common family groups. I started with just 4 groups for each of my grandparents, and then refined the groups from there. This is a very slow process, and I still add more as more people add their DNA and appear on the match list. It's very simple. All we need to do to start with is choose a color for each of our 4 grandparents and begin grouping the matches into those groups. So for example here, if I knew that Craig related to my grandmother on my father's side, I chose the Winter Wachowski group and made it green. And so I add him to that group by clicking the little arrow. But to add other people to the Wachowski Winter group, I click on Craig and go to his shared matches. Because anyone who shares DNA with my father and with Craig, who's on the Winter side, they must also be on the Winter side. So now I just run down the list and click everyone green as being a Winter Wachowski side relative. Obviously, I've got all my dad's DNA, so I come up as a match. So if I wanted to add my group, I'd just click Add to Group, choose Wachowski, the green, and Add to Group, and I would be on it. But if I had not known that Glenn was related on the Winter side, if there was no tree and I did not know who he was, immediately he shares DNA with Craig and my dad, so I know he's on the Winter side, so I can put him in the Winter group. And similarly, with everyone under the shared matches, with this person and my dad has to be in the green group. We then go back to my dad's matches and choose the next group. If I know that Dennis is on the Roberts-Rowland side and I've marked him red, then if I look at his shared matches, everyone on the shared matches will be on that side of the tree, so I'll mark them all red. And then very soon we will have everyone grouped into the groups of our 4 grandparents. We can then move on and choose colors for our great-grandparents and start breaking the groups even smaller. Here again is our close family tree, starting with my father, his parents, grandparents, and great-grandparents. And with the DNA matches, I'd group them into the Pochnik group, the Wachowski group, and the Brousseau group. And all the rest would be the Rowland-Roberts group. Some in this group were descendant of Richard Charles Roberts' first wife in South Africa, and others were close relatives, seconds or third cousins, not descended from his first or his second wife in South Africa. So these were going to be investigated next. Finally, I had my dad's DNA results. The ethnicity estimates were surprising because although there was the expected 50% Austro-Hungarian, 25% roughly German, there was a surprising amount of Italian DNA, and we had no idea where that came from, and a very small amount of English DNA. Then on the matches, I started grouping them, and once I had separated out the Roberts-Rowland group, I found that there were some very close second and third cousins who lived in other parts of the world, in Australia and Chicago and Massachusetts. I had no understanding how they could be so closely related to my father when his ancestors had moved to Africa when they did. The next exciting part of the investigation involved contacting these close relatives to try and find out what their trees looked like and how they could be related.